G'day everyone, my name's Rob Smith. I'm going to be presenting this session on the structural connectivity aspect of brain network analysis. Now this presentation is going to be slightly different in its construction rather than simply listing all of the different tools available. I prefer to try to convey a deeper understanding of exactly what we're doing with these technologies and kind of give people the power to critically analyze what they see in the literature and make decisions around their own experiments. So I'm going to start with a very quick overview of the basic framework for structural connectivity estimation. I'll talk a little bit about the streamlines tractography paradigm. I'm then going to frame Connectome construction in actually a much simpler context of this quantification of connectivity. And then I'm going to go over this list of kind of requirements that I've devised over years of thinking about this stuff, of ways to critically analyze whether or not the requirements for performing robust Connectome construction are actually satisfied. So we'll start with simply the basic of the paradigm. So obviously we're focused on the white matter with diffusion MRI. If we've got a particular white matter bundle of interest, we do diffusion MRI, we do some amount of pre-processing, which is unfortunately beyond the scope of this particular course. And from that, you have your conditioned DWI data. You're going to fit some kind of diffusion model to your voxel-wise diffusion signal, and that's going to give you a model fit in every voxel. In this particular case, the intensity of the image reflects the estimated white matter fiber density. And in addition to that, in every voxel in the image, we have an estimate of the fiber orientations and densities. Now from this, we're obviously going to perform tractography, which we'll speak about in more detail later on. That's what gives us the actual reconstructed trajectories through three-dimensional space. And ultimately what we want to be doing is somehow utilizing this reconstruction to derive some measure of connectivity. And ideally this measure is going to be going to tell us something useful about the actual underlying biology. But this again, later on in the talk, I'll delve into more detail as to exactly what we want / can quantify in this aspect. So moving on to streamlines tractography. This was proposed by a few different people in about 1999, 2000. Realistically, the underlying paradigm hasn't actually changed for about 20 years. In comparison to what most neuroscientific applications are using now, there aren't actually tremendous differences. But one key thing to focus on here is the word streamlines. That'll become important in a moment. And the basic premise is very simple. You have a voxel grid. In every voxel, you have your diffusion model fit that's going to provide you with one or more estimates of fiber orientations. And the goal here is to simply move along in a stepwise fashion along the orientations of these fiber direction estimates. And it actually doesn't get a whole lot more complex than that. When you start looking a bit more into the pragmatics of how a streamlines algorithm works, here I've broken it into 4 different sections. One is what I call streamline seeding. So this is simply determining where in space you're going to start your reconstruction. There's the way in which you sample from these fiber orientations in order to propagate the streamline. You then have to, at some point, make a decision about when you are going to stop propagating the streamline to terminate it. And then depending on your experiment, you'll have some sort of criteria that define whether this is an acceptable streamline or whether it should not be used. And these can be things like minimum or maximum length, constraints on a priori conditions of where streamlines are or are not allowed to go. Again, very, very context dependent on your particular experiment. A key little point to bear in mind here, particularly in the context of this course, dealing with network analysis more generally, is that although the actual streamlines generation process is explicitly directional, once you've got that streamlines reconstruction, it should be considered undirected. And ideally, if the reconstruction algorithm is robust, you should be getting approximately the same answer regardless of which direction you actually generate the streamline in. Just to give you an idea of, I've said that streamlines algorithms are very simple, conceptually, pragmatically, but again, depending on exactly what you're doing, there's a huge number of underlying variables that don't necessarily have an unambiguous, sensible default to them. And for a lot of them, changing values is going to drastically change the outcome of the reconstruction. So what I've got here is the help page of a software command responsible for generating streamlines. And as you can see, the length of this help page is absolutely enormous. And this just reflects the flexibility, but also ambiguity in how to, the scope of the tools that are available to you when you're performing streamlines reconstruction. And finally, a quick mention on there are forms of tractography that are not streamlines tractography. And I sometimes see this mixed up in communication. So I just wanted to be very clear that although streamlines is a form of tractography, it's not the only one. There are various other techniques that deal with front evolution. Geodesics involve constraining your endpoints and then looking for the most probable path between them. And there's also various different types of tractography algorithms that try to simultaneously optimize the entire reconstruction as opposed to doing each connection in a sequential fashion. But when it comes to neuroscientific applications, the vast majority of applications are using streamlines algorithms. Okay, so now I want to talk about the way that I conceptualize and kind of break down connectome construction, a bit of a divide and conquer strategy. And this is basically what we're doing is that we have a bunch of pathways of interest. Now, when it comes to connectome construction, this typically means you have a pair of grey matter nodes and you want to quantify the connectivity between those two specific nodes. So what we have here is this kind of sub-experiment where we are identifying or reconstructing a specific pathway of interest. We're going to derive from that reconstruction some quantity of connectivity. And then the process of connectome construction is simply repeating that process over for every single possible pair of grey matter regions. And then obviously if you're attending a network analysis course, you're probably interested in then what you're going to do with this data. But from the perspective of this particular talk of quantifying structural connectivity, I really want to focus on this this first idea here of what we're actually extracting and quantifying. So here on the left, I have a very coarse parcellation of brain grey matter just into 4 lobes per hemisphere. On the right, I have a whole brain streamlines tractography reconstruction. And ideally, we want to somehow combine these two things together to produce a matrix, the connectome matrix. And obviously what this matrix actually represents is if you look in any particular element within this matrix, the intensity or the value stored here reflects some measure of connectivity. So this entry here corresponds to the connections between the left and right frontal lobes. Here's the resulting streamlines. And we can do the same kind of visualization for every entry in this 8x8 matrix. But the point is that the actual process of identifying and quantifying each of these pathways is the same thing, just repeated over and over. So here's actually what it looks like if you try to visualize every edge in the connectome at once. But as I mentioned, the key thing I want to do here is show that for the sake of genuinely understanding the framework of structural connectome construction, you really want to focus on this idea of, okay, I have a pathway, a white matter pathway of interest. I want to quantify some measure of connectivity. If you can get that right, then you can get connectome construction right. And so for the subsequent section, that's what I'm going to focus on, is these kind of fundamental requirements that you need to satisfy during this process of quantifying white matter pathways. And this comes partly from the progressively increasing complexity of both the state-of-the-art methods that are developed and the various software packages that are made available, the huge number of options that are available to you. It's very difficult to give a course here where I would simply say, okay, use this software and use this algorithm and use these parameters. And that also wouldn't be of much assistance when you're reading through the literature and you want to critically assess whether or not you trust the results that someone has generated based on the techniques that they've used. So what I want to do here is convey some ideas that I've been formulating in my head over the last few years of trying to break down all of these concepts into their most fundamental form. And that gives you a novel power to really critically assess these things. And it also means that when you come across a new method that's got some fancy acronym associated with it, you're better positioned to appropriately place exactly where such methods fit within the overall framework of the reconstruction and quantification framework, because otherwise you get a kind of blurring of different concepts. So I've managed to, in the process of this thought experiment, to generate this acronym track, and this is going to be these four fundamental requirements. So the first one is trajectories. Fairly simple. If you are doing a digital reconstruction of white matter pathways, if the trajectories you reconstruct are not reflective of the biology, then you're kind of wasting your time. Seems fairly obvious, but I'm going to break it down a little bit more. First thing I'm going to do is I'm going to split this into the estimation of fiber orientations and then the utilization of those orientations during tractography. So the role of estimating fiber orientations, this is the responsibility of what we refer to as the diffusion model. So this is the model to which you provide the diffusion weighted signal intensities for a given voxel, and it yields hopefully some information with respect to fiber orientations, densities, or other relevant parameters. Now, obviously, with Streamline's tractography, it's this iterative process where we're sampling these orientations multiple times for each reconstructed streamline. So obviously, you need for these orientations to be accurate at every single step. It only takes one single incorrectly estimated orientation for your streamline to go in completely the wrong direction. So when comparing different diffusion models, we can think about different attributes that we want our diffusion model to have in order for it to be sufficiently robust for tractography. One is that we want to actually yield estimates of fiber orientations. This is slightly a nuanced distinction between diffusion orientations. I won't bore you with that. A big one here is that you really need to be using a model that can resolve multiple fiber orientations in each image voxel. And this is very frequently referred to as the crossing fiber problem, which is basically that if you look at the first proposed, well, it's not quite the first proposed model, but the first proposed model that could provide these orientations was the diffusion tensor model. And as long as you've got a single coherent bundle of oriented fibers in each voxel, the diffusion tensor does a pretty good job of telling you what orientation those fibers are in. But as soon as you get to the stage where you have multiple different fiber orientations within a single image voxel, the diffusion tensor starts to no longer provide useful information. So ideally you want a what's sometimes referred to as higher order models. I'm showing one specifically called CSD in this case, but there's a huge list of these different models. Ideally, you want something that actually tells you the different orientations of the different fiber bundles in each voxel. ideally, you would want to have some measure of the underlying dispersion of those fiber orientations. There are some models that can do this, but not many. There's a whole issue of the amount of uncertainty within your data. There's also an issue of how do you determine the orientation in which to propagate your streamline if the streamline position is not right at the middle of a voxel, but it's kind of in between two voxels. Now, obviously this is a long list, and there's unfortunately no single model that can provide you with all of the desirable attributes. I've listed my kind of subjective classifications for a few common models here. Unfortunately can't go into all of the details here. It's just worth bearing in mind that, yeah, when you're assessing a particular model critically for use in tractography, here's a few different attributes that you can assess critically. So secondly, following the local orientations, this is obviously the role of the particular streamlines tractography algorithm that you're using. And basic premise is you've got a streamline, you're at a particular vertex in three-dimensional space, and you need to determine the direction in which the streamline is going to propagate. So a few things that are typically referred to in this context is you'll have some kind of angular threshold. You don't want streamlines to bend too much. You don't want them to instantaneously loop back upon themselves. And you'll regularly see referred to the step size. That's simply the distance in 3D space between the vertices. Now, as I mentioned previously, one of the issues we have with streamlines tractography is we have all these parameters and we kind of don't quite know what to set them to. Obviously, from the perspective of providing research softwares, people will provide what they deem to be the best kind of defaults they can, but they don't always necessarily apply to all data. And there are obviously pros and cons of tuning these things. So this is a great little paper. It's quite old now, but simply visualizing the kind of effect that these basic parameters can have on the outcomes of the streamlines reconstruction. Another common parameter within the algorithm is some kind of threshold on the diffusion model to determine where streamlines should start and where streamlines should stop. And obviously, if this threshold is very high, your streamlines can often terminate before they've completed reconstructing your pathway of interest. Whereas if it's too small, your reconstruction can be dominated by noise. Another little hidden one is what's referred to as the order of integration. This has to do with how the algorithm behaves in the presence of curvature. Generally, a higher number is better. You want a second order method or a fourth order method over a first order method. It's a little bit more complicated than that, but this is at least a demonstration of the kind of effect that this algorithm attribute has. Quite often you'll see this distinction of deterministic versus probabilistic algorithms. It's often described that there are two types of streamlines algorithms, deterministic and probabilistic. I find that a bit too reductionist. This is just one of many features that these algorithms have. Basic premise is that if you generate more than one streamline from exactly the same starting point, do you always get the same answer? So that's a deterministic algorithm. Or do you get a different answer each time? And that's a probabilistic algorithm. And the debate about these two approaches is still ongoing. There's definitely no consensus. So the second one is, again, the second point of these four is again quite easy. It's reconstruction density. The basic point here is that you need to have a sufficiently dense reconstruction so that whatever quantity you're deriving from your experiment is reproducible. And to greatly simplify this expression, it's the old question: how many streamlines should I generate? This question comes up all the time for Connectome construction. Unfortunately, we still don't particularly have robust answers on this. There's a few people working on various experiments for it. The way that I like to conceptualize it personally is that if you generate some number of streamlines, you produce your Connectome, you do an analysis. If you then regenerate that same number of streamlines, regenerate your connectome, redo your connectome analysis, and you get a completely different answer, then you don't have enough streamlines. If you're generating enough streamlines, that process should, to within some precision, produce the same result. Now, obviously, the converse point to that is that you don't want to be waiting for a month per subject for your streamlines to generate. So there is some degree of balance there, but it's worth thinking about critically, or even potentially performing that experiment yourself rather than hoping that someone will give you a number. And this inevitably depends on the particular experiment that you're doing. In the context of connectome construction, it might be how many different nodes you have, that sort of thing. This is just a quick little reference. So this is not connectome construction, but this is purely looking at estimating the volume of a particular white matter bundle. And as you generate more and more streamlines, the volume persistently goes up because there are always new probabilistic streamlines finding some voxel that no other streamline had been to yet. So obviously, if you're trying to quantify volume and say compare it across subjects, this is an effect that you need to be aware of and think long and hard about how you might deal with it. In the context of connectome construction, I tend to think a fairly big influencer here is the number of nodes that you have in your parcellation. If you have 10 nodes, you might have the 4 lobes of each hemisphere plus your 2 cerebellar hemispheres. The number of streamlines you need to estimate that connectivity robustly is probably not as many as you would need as if you've got a super fine parcellation. So these are entirely back of the envelope numbers. This is simply a demonstration that if you think about, okay, the number of edges in the connectome goes up as the square of the number of nodes. So as you increase the number of nodes in your parcellation, there's a good chance that the number of required streamlines is going to grow very quickly. Okay, so on to the third of 4. This is one slightly more cryptic. It's what I'm calling attribution. And basically the idea here is that you have, you're doing a streamlines reconstruction. You've got your grey matter parcels defined that say, which pathways that you want to quantify. You need some process by which to say, which of these streamlines, either how do I generate streamlines or which of these pre-generated streamlines do I select that belong to each node, belong to each edge in the connectome. So this again is one that I'm going to break into 2. I'm going to talk about terminating and then assignment of streamlines. So when it comes to the termination of streamlines, a long-standing issue with streamlines algorithms is that it's very hard, while your diffusion model will tell you the orientations of the underlying fibers, it's not very good at telling you where they start and where they stop. Historically, people use things like some kind of mask image. You might apply some threshold on the amount of fiber anisotropy in your diffusion model. There might be something to do with curvature. And what this image on the right shows that it's possible for there to be multiple different configurations of the underlying biological fibers, all of which lead to the same diffusion weighted signal. And yet some of these involve fibers that go all the way through, some involve fibers that terminate. So this is historically very ill posed. Generally, the best way to do it is to make use of high resolution anatomical contrast image. So this can be in the format of a native surface representation of the cortex, or it can be image based. But the key thing here is that you don't just want to constrain where the streamlines go, where the streams are allowed to go, but also where they have to stop. So each of these little yellow dots is a streamline termination. And you can see that if you use only the diffusion image data, the streamlines kind of start and stop all over the place. It's all very messy. Ideally, what you want is you want to engage some method that will more carefully constrain where the streamlines are forced to stop, and also where they don't stop. You don't want streamlines going into your lateral ventricles, for instance. And obviously, if you're using high resolution image in conjunction with your diffusion image data, the correction of susceptibility distortions in your diffusion data is paramount. Okay, so assigning streamlines, this has been a little bit of a bugbear of mine that a lot of people overlook. If you've got a grey matter parcellation and you have a whole brain streamlines reconstruction, there needs to be some explicit process by which you combine those two things together to produce the connectome. And this is very often not reported in the literature, but it can potentially have large consequences. So we actually published something on this not too long ago. And yeah, the basic premise is that depending on the particular mechanism that you use for assigning streamlines to parcels, depending on the nature of your streamlines reconstruction, depending on the nature of the parcellation itself, you can get quite drastically different results. So I won't go into full details on all of the different options here. It's more a question of if you're doing structural connectome construction, it's worth being aware that there is actually a step required here. It's not something that happens magically. Okay, so the fourth requirement in my list is quantification. And this is basically, okay, we're generating a structural connectome. We are wanting to quantify connectivity between regions so that we can do some analysis on it. But what are we actually quantifying? So this again, I'm going to break into two different parts. I'm going to focus mostly on what I'm calling here intra-subject. So the basic question here, at least in the context of connectome construction, is okay. We've got a parcellation. We've got a set of nodes. We've got all of our edges between our different nodes. What are we going to quantify for each of those edges? When we say that we're quantifying structural connectivity, there's a slight ambiguity in that statement. And there are, you can use your fancy software, you can do your reconstruction, you can do all your fancy visualizations, but ultimately what you've got here is a couple of grey matter parcels, you've got streamlines between them, and you want to derive from this experiment some measure of connectivity. And ideally that measure of connectivity needs to be robust, but it also needs to make sense in the context of what you're then going to be doing with the connectomes that you've produced. So historically, and probably still the most frequently used measure is simply the number of reconstructed streamlines. This has again been a bugbear of the more hardcore developer community, but it is still something that you see used a lot throughout the literature. I don't have the time to go into full details here, but there are a number of good review articles that talk about this. Another common one is to do something like the mean fractional anisotropy. So just quickly, this FA measure is derived from the diffusion tensor model. It's a measure of whether the diffusion signal is very anisotropic or whether it's more kind of ball-shaped isotropic. So the basic idea here is that you've done your streamlines reconstruction of your connectome edge. You can use that to generate a spatial map of streamlines density. You might apply a mask to this to produce a voxel mask. You can then take your quantitative image. So this is the fractional anisotropy measure here. And so you might apply your streamlines mask to that image so that you can see only the FA values within that mask, and you might then take the mean of that. So this is another very common measure used. one of the big issues with it is that all of the weaknesses that I described previously with respect to the diffusion tensor model failing to resolve crossing fibers, that limitation propagates up to the meaningfulness of the FA measure. You can also do the same type of quantification, but not using specifically the fractional anisotropy measure. You might use some other type of image-based quantitative measure. So basically what's happening here is you're taking a quantitative image and kind of projecting that up into the space of your connectome edges. So there can be an issue there if you've got a voxel-wise quantitative measure, but have performed tractography including crossing fibers. You have the effect where a genuine biological effect in one crossing fiber bundle is going to influence what you quantify in the crossing fiber bundle. There's also the further issue, which kind of depends on the type of connectome analysis you're going to be doing. Quantifying these kinds of values within a connectome is absolutely fine. The question is, if you're going to be doing some kind of fancy graph theoretical analysis, it needs to make sense for whatever quantity you're deriving on a per connectome edge basis to have meaning within the process of that connectome analysis. Sorry, I was supposed to jump to that slide earlier. Personally, when I think about graph theory analyses and what they're actually doing, I think about, okay, we're trying to quantify the transfer of information around different nodes in the network. So what quantity would we ideally want in order to properly capture that? This has been some of the work that I've been doing myself historically. The basic idea here is that we want some measure of kind of bandwidth, the information carrying capacity between different nodes of the network. And the way that we're going to at least try to quantify this is by capturing the intracellular cross-sectional area of fibers. So this is what this actually looks like. If you can imagine that these two boxes at the ends here are your grey matter nodes. You've got some, in this case, biological fiber bundle between them. If you take a cross-section through that bundle and then look at the intracellular cross-sectional area here, this is something that, at least within limitations, we can try to quantify, specifically because there are a lot of diffusion models that we give us estimates of fiber intracellular volumes. So this is the quantity that we've been trying to get at, and the basic theory behind how it works is that if you have an image where you have your fiber orientation estimates, if you do a basic whole brain streamlines reconstruction on that data, and then generate a map of the distribution of streamlines density throughout the image, you can see there's a discordance between the distribution of white matter fiber density and streamlines density. And this is one of the contributing factors towards the common criticism that streamlines cannot be used quantitatively. So the goal of, in this case, I'm showing the SIF2 method, but there are a couple of other methods that do very similar thing, is that it modulates up or down the amount of contribution that each streamline makes towards the quantification of connectivity. And that optimization is done in such a way that you begin to achieve this concordance between kind of modulated streamlines density and underlying white matter fiber density. And that's what, by applying one of these methods, it provides a little bit more biological relevance and quantitative properties to what would otherwise be streamline count. So finally, a little note on inter-subject quantification. And this one's really more of a quick little thought experiment at the end. If you are not actually doing any kind of advanced graph theory analysis, but you simply want to take the raw connectivity values within the edges of the connectome and compare those across subjects. The question is, can you actually do that comparison naively? Now, if you're extracting something like the FA measure, that measure has a kind of intrinsic normalization, which means if you calculate FA in one subject, you calculate FA in the other subject, you can compare those two things. With something like streamline count, it can be a little bit more nuanced, and it depends on the particulars of how you actually do your connectome construction. But it's nevertheless something worth bearing in mind of, okay, if there are various mechanisms by which these numbers may vary across subjects, is it reflective of biology, or is it some kind of reconstruction bias, that sort of thing? This measure of fiber intracellular cross-sectional area has its own technique that I won't bore you with. But it's also worth thinking about these global network measures or other higher order analyses that people do with connectome data. And the way I like to think about this is, say you take your matrix data from your reconstruction and you then simply double every value in the matrix. If the result that comes out of your analysis is different, then there's scope for any influence that differs between subjects that modulates that kind of global amount of connectivity to potentially be driving differences in your analysis. So you need to think about, is there scope for some effect that I'm not interested in to be driving differences in my actual derived quantity of interest? Again, more of a thought experiment. So that's it for me. That's again just kind of showing the basic overview. I hope that formulating this particular educational session in this way provides some insight, even if it's not a step-by-step instruction guide. Obviously, I'm contactable by email. I personally work on the Matrix software, and we do have a fairly active community forum now where people are free to ask questions about all this sorts of stuff. I'll leave it at that. Thank you.
So welcome to the fifth session of the Network Neuroscience Education Workshop. My name's Andrew Zaleski, and I'll be presenting on connectome statistics. I'm going to begin with introduction to network thresholding. I'll then move on to null models, and finally I'll wrap up with comparing connectomes. I'll particularly look at connectome inference and connectome prediction. All these slides can be downloaded from the website that I've shown here on the first slide. And if you have any questions, please don't hesitate to send through an email. So connectome thresholding, what is thresholding? Well, thresholding is performed immediately after the connectome has been mapped, whether it be a structural or a functional network. And it simply involves removing the weakest connections from the network. And this can be done in an incremental way, and it's governed by, and it continues until we've reached our desired connection density. It's important to emphasize that thresholding is not essential, but it can help in terms of improving connectome visualization, reducing false positives so that the weakest connections are more likely to be the connections that are spurious. It can simplify the interpretation and computation of some graph analyses, particularly when we're computing null networks. For high dimensional networks, so when we're mapping networks at the level of vertices or voxels, it can reduce storage burden. And finally, some recent work by Buchanan and colleagues has shown that thresholding can in fact strengthen associations with age and behavior. Now, the counter argument is, you might want to consider, why not to threshold? And as I said, thresholding is not an essential step. And some work by Sivia and colleagues has shown that, first of all, the thresholding cutoff value is arbitrary. So should we threshold at 10%, 20%, 30%? It's not clear. It's generally an arbitrary choice. Moreover, the argument that Sivia and colleagues make is that the connections that we throw away with thresholding are generally the weakest connections. And so it turns out that whether or not we throw these connections away, their overall influence on network measures, such as path length in this example, is probably negligible. So they contend that, well, what's the point of including a thresholding step if the overall consequence is minimal? There is one further step that can be done. Once we've thresholded our network, we can also then go on and binarize the network. And this is where we're only interested in knowing whether a connection is present or absent between a pair of nodes. We throw away the information about connectivity strength, whether that be functional, the correlation coefficient, or the streamline count. The disadvantage of binarization is that this variation in connectivity strength is most likely to be informative and biologically meaningful. We see a rich variation across pairs of regions in connection strength. However, the counter argument is that it's probably likely that some of this variation is due to noise or methodological confounds, and therefore by binarizing connections, we potentially remove some of those confounds. So here's a conceptual example of thresholding. We start here. This network has three types of connections: weak, moderate, and strong. And so in the first step, our network comprises connection density of 26%. Connection density is just the actual number of connections in our network divided by the total possible number of connections. So in the first step here, we eliminate the weakest connections, and this reduces the connection density to 20%. Then we can go one step further and also eliminate not only the weak connections, but also the moderate ones. And this further reduces the connection density down to 30% in this example. But note here that one of the nodes becomes fragmented from the network. And this can happen if we threshold too severely. And this fragmented node can be problematic for some graph analyses. There are two broad classes of thresholding methods. The one that I've shown here up on the top, which is referred to as density thresholding, is where we prescribe a desired connection density, in this case 20%, and we remove the weakest connections until we achieve our desired connection density. And this is achieved with the brain connectivity toolbox function called threshold proportional. The other kind of thresholding method is weight-based thresholding. And in this case, rather than prescribing a connection density, we prescribe a minimum weight. And any connection that's below that minimum weight is discarded. So in this case, we say any connection, an example that I've shown here, any connection that comprises fewer than 3 streamlines is discarded. So we see that the connection here between comprising 2 streamlines and 1 streamlines is removed. The crucial thing to note here is that there's a one-to-one correspondence between density and weight-based thresholding. For any given density, I can tell you the equivalent weight that will achieve that density, and it goes the opposite way as well. So you might argue then, well, what's the point of if there's a one-to-one correspondence? Why should we have these two different thresholding methods? And this difference comes about when we look at groups of subjects. And in this case, in this example, I've shown 2 connectomes, 2 networks, Jack and Alice, and we see that Jack's connectome comprises a greater number of strong connections. These are the edges, the thicker edges that I've shown in the network. Now, if we threshold in such a way that we discard the weak and the moderate connections, we see that Jack's thresholded network comprises a greater number of connections compared to Alice's network, to Alice's thresholded network. So then if we go on and do subsequent graph analysis, network analyses on these thresholded connectomes, any differences, for example, in path length or clustering coefficient between Jack and Alice are most likely driven by this confound in connection density, namely the confound in the different number of connections. So you might say, well, given this confound, maybe we should be using density-based thresholding. And by definition, density-based thresholding ensures that both Jack and Alice's thresholded network comprise exactly the same connection density. In other words, the same number of connections, 7 connections in both cases. But the disadvantage here is that in the case of Alice, to get Alice's network to 20%, we've had to admit some of the connections with moderate strength. And these connections are more likely to be spurious. So we're kind of adding in connections to make up the numbers, but these spurious connections can add noise and a different kind of confound to the network. This difference between weight and density-based thresholding comes up in patient control comparisons. And so this is an example in schizophrenia. In general, what we see in schizophrenia, functional connectivity is weaker in patients compared to healthy controls. So if we apply a weight-based threshold, so if we remove functional connections with a correlation below 0.2, we see that there's a striking difference in connection density between the patient and the healthy control, 53 by 75. And this would confound any subsequent network analyses, potentially confound. If we use density thresholding, sure, we guarantee that there's a matching in the connection density, in the total number of connections between the patient and the control, but we've had to lower the bar. We've had to lower the bar on the minimum connection weight for the patient. So we have to drop that down to 0.31. And so the additional connections that we're adding in could potentially be spurious connections, false positives, which can add noise to the thresholded connectome. Another key consideration is the choice of threshold. Should we threshold at 5%, 10%, 20%? It's not clear. And so to get around the dependency of threshold, it's recommended to evaluate, to perform the analysis, the network analysis across a range of thresholds, and then perform statistics on the area under the curve. So in this case, we've mapped path length as a function of connection density and then computed the area under that curve. This also relates to topological data analysis. Check out their work by Anne Sizemore. There is also an approach called multi-threshold permutation correction. And this is where we look, it's also evaluating a range of thresholds and looking for a continuous range at which we can reject a null hypothesis. This is work by Drake Smith and colleagues. A more recent thresholding approach is referred to as consensus thresholding. This is where we rather than thresholding individuals, we look for a consensus across a group of individuals. And in this case, we see that in Alice's network, if we look at this particular pair of connection, this connection here, we see that that's a weak connection, but it survives in the network. And the reason why it survives is because Bob and Jack, that connection is actually strong. So we need that there be a moderate or strong connection in 2 of the 3 individuals for the connection to survive. In what I've discussed so far, thresholding has been performed on connectivity strength. An alternative approach is to cast thresholding in a statistical framework. In the case of functional connectivity, we can convert the correlation coefficient to a z-score, and then that z-score can be we can convert that to a p-value, and thresholding can be performed on the p-values in a statistically principled way. The tricky thing here is that due to autocorrelation in the ball time series, the degrees of freedom are often inflated, and therefore our p-values are optimistic. Afayuni and colleagues have proposed an adjusted z-score where they estimate a more realistic degrees of freedom and yield a more accurate p-values. Check out the work if you're interested in thresholding p-values. Another approach is local thresholding. So what I've discussed here are global thresholding methods where we apply one global threshold across the whole network. There can, however, be locales within a network where a connection is strong relative to its neighboring connections, but within the global network it's weak, and we might want to maintain those connections. And so there's approaches such as the disparity filter that can allow us to do this. The other key concern is if we threshold too severely, nodes can fragment. So there's approaches such as the minimum spanning tree, which will guarantee against node fragmentation. So in summary, I've introduced connectome thresholding. I've mentioned that this is It's not an essential step, but it can alleviate false positives, ease the computational burden, help with connectome visualization and the interpretation of some measures. I discussed the two most common thresholding methods, those being weight and density thresholding. I also highlighted the importance of evaluating a range of thresholds. And so if you're interested in actually doing this on your own data, the BCT functions for weight and density thresholding are shown here. So in the next part of the talk, I want to move to null models. And null models are crucial in connectomics to set references, to set benchmarks. And I want to begin with this example. So this is a small network, and I want to consider the network measure of path length. We'll see this again in one of the subsequent sessions of this workshop. So in this small network, the average path length is 2. So this means to get between any pair of nodes, we need to traverse 2 connections, as I've shown here for the particular example. And so the question is this, if you map a connectome and you find that the average path length is 2, is this long, is it short, or is it typical? We don't know. It's hard to determine just based on a measure of path length how to interpret that. And so we need some kind of benchmark. The simplest benchmark is to compute path length in a random network with the same number of nodes and edges. So there's a closed form formula to do that. We can plug in the number of nodes, 9, and the number of edges, which is 15. And we find that for a random network, the average path length is also 2. So this suggests that for this particular network, there's nothing unusual. There's nothing that's particularly conducive to efficient routing of paths. The path length is actually exactly the same as what we would expect in a random network. Let's now move on to this network. This is a bigger network. This is a lattice network. And we see that the average path length is 2.5. So we have to traverse 2.5 connections to get between any pair of nodes. And we ask the same question, is 2.5 long, is it short, or is it average? And so we can once again benchmark to an average network. We plug in the number of nodes, the number of edges, being 20 and 40 into this formula. And we see that the average path length for a network with 20 nodes and 40 connections is 2. It's actually shorter. So what we can say is that a lattice is not particularly conducive to efficient routing, to efficient shortest paths. There's something specific about this topology that makes it not efficient for routing. And this is a well-known fact about lattice networks. So in this example, the null network, the reference network, is the random network that's matched in the number of nodes and edges. But you might think, well, what other things might we want to match? Do we really just want to match the nodes and the edges? We might need to match the node degree, the connection length, and so on and so forth. So we can consider a nested family of null networks. And so in the outermost circle, we have the space of all possible networks. And this is probably not a very useful null network. And in the innermost circle, we say we want to match the number of nodes to our actual connectome. Then we say we want to match not only the number of nodes, but also the number of nodes and edges. And this is exactly what we did in the previous example. We had the closed formula, the closed form formula that gave us the path length for a random network with the same number of nodes and the same number of edges as our actual network, as our actual connectome. But we can go further. We can also not only match the number of nodes and edges, we can also map node degree. And this is perhaps the most common type of null network in connectomics. That's the green family of null networks. And more recently, there has been endeavors to also match the geometry of the connectome. And this gives us the innermost circle here. So we're gradually adding more and richer properties to our null, which confines the null more and more. Of course, if we add too many, if we keep adding properties, we're eventually going to converge to our real network, to our actual connectome. So how do we actually do inference with these kind of null networks? A common approach is that we sample null networks from this null space. So in here, I've shown the most common family of nulls where we match the number of nodes, number of edges, and the node degree. And the purple crosses here represent random networks that match these 3 properties. So we can generate tens, 20, hundreds of random networks as such. We can compute path length in each of these networks, and then we can divide that by the actual path length in our real connectome. And so if this ratio is 1, it says that our actual connectome, the path length is not particularly unusual. In contrast, if it's greater than 1, it suggests that our path length is exceptionally long relative to a random network that's matched in terms of these 3 properties. We can also compute p-values to assess the same hypothesis. So you might ask, how do we actually generate random networks? How do we sample networks that preserve the number of edges, the number of nodes, and the node degree? And we can do this very efficiently with Maslov-Sneppen, with the Maslov-Sneppen rewiring algorithm. This involves taking our connectome, we identify 4 nodes that are connected in the pattern shown here. So U is connected to V and I to J. And we then rewire them such that U then connects to I and V connects to J. And we repeat this step again and again. So we keep finding 4 node motifs, such as the one shown here, and rewiring them as such until we reach an adequate level of randomization in our network. And this gives us 1 sample from that null space. And we can do that again. And so we can generate a family of null networks, and we can compute our network measure in each member of that family. And this enables us to build up an empirical null distribution. It is a little more difficult to do this for unthresholded weighted networks, but check out the work of Rubinov and Sporns for some extensions of Maslov-Sneppen rewiring in those cases. So here's some examples of Maslov-Sneppen applied to real-world networks, to a macaque and a mouse connectome. There's a sample of the Maslov-Sneppen network that's shown here on the right for both of those connectomes. In practice, we generate hundreds of such samples. And here we're showing the normalized clustering and path length. So you see that the normalized clustering is greater than 1 relative to, so this is what we've normalized by the Maslov-Sneppen rewired. So it suggests that the connectome is clustered to a greater extent than what we would expect in a random network. In contrast, the path length is exactly 1. The normalized path length is 1. So it's saying that the path length is what we'd expect in a random network, and small worldness is greater than 1. So these are small world connectomes of small world networks, which is a well-established fact. More recently, there has been interest in incorporating geometry into our null networks in connectomics, and this allows us to assess the impact of geometry on network topology. So Roberts and colleagues have developed an efficient approach to preserve geometry in our null networks. Basically involves regress. So geometry is contextualized in terms of fiber length, the distance, the physical distance between pairs of regions. And the first step is to regress out this fiber length. We then shuffle the connections and then basically add the geometry back into the shuffle connections. And this generates a realization, a sample from the null network. So what we see here is once we add geometry back into our null network, if you focus on the plot here on the bottom, which is clustering coefficient, this is the blue curve, the blue line is the clustering coefficient for the real connectome. The red here is for the non-geometry preserving, in other words, Maslov-Sneppen rewiring, and there's quite a big gap between the real connectome and the red line. In contrast, once we add geometry back into our null model, we see that the clustering coefficient it becomes closer to what it is in our real connectome. So this suggests that geometry is influencing, at least to a certain extent, network topology, in this case called the clustering coefficient. There is an alternative or a related concept in null models called generative models, and this is an idea where we grow networks in silico according to particular wiring rules. The idea is that we grow the networks and then compare them to the empirical connectomes to see how well they match. So Petrovertis has considered 3 different models. There's a geometric component to the model, and then there's a topological component. The best fitting model was the one that's shown on the bottom here, the economical clustering. So Pij represents the probability of forming a connection between a pair of nodes. So the best model was that we encourage connections between pairs of nodes that are close to each other, that are physically close to each other, and also that share common neighbors. And this is a property referred to as homophily. Rick Betzel also generalized this model to include other topological rules. Once again, the best fitting model was one that included a geometric constraint, promoting connections between pairs of nodes that are close to each other, and also a homophily constraint. So in other words, encouraging connections between pairs of nodes that share the same neighbors. So in summary, I've shown that null models can set expectations for network organization under certain null hypotheses. The most common null network involves preservation of the number of nodes, the number of edges, and node degree. And this can be efficiently realized with the Maslov-Sneppen rewiring algorithm. In recent times, there's been interest in also preserving not only the edges, nodes, and the node degree, but also the geometry of the connectome. This is often contextualized in terms of fiber length, connection length, in other words. And I've also covered the related concept of generative models and shown that both geometry and homophily play a key role in evolving connectomes in silico. So if you're interested, the BCT functions for Maslov-Sneppen rewiring and generative modeling are provided on the bottom there. So in the final part of this, this session, I want to move on to comparing connectomes. In other words, connectome statistics. And so the idea here is that you may have mapped connectomes in a group of healthy controls. You may have also done the same thing for a patient group or a treatment group. And the aim then is to identify connectome attributes that differ between the control group and the patient group, or that are associated with a certain behavior or are modulated by treatment. An alternative goal might be connectome-based prediction, and this is where we want to actually use connectome attributes to predict something about the future. That might be clinical outcome or response to the treatment. So I'm going to consider both connectome inference as well as connectome prediction. In both these cases, the challenge is the high dimensionality of the connectome. We're typically dealing with hundreds of nodes and potentially thousands, tens of thousands of distinct connections. So what are the different attributes that we can compare between connectomes? The simplest is we can do a global comparison. So each individual is characterized by one value, one summary value that characterizes their connectome. This can be something, a summary value such as efficiency, modularity, Rich-Cobb coefficient. The disadvantage of this is that it doesn't give us much localizing power. If we find a difference in something like efficiency, it doesn't tell us what region, it doesn't tell us what particular connection is associated with that difference. We then move to node level comparison, which gives us a little more localizing specificity. This is where we test hypotheses about particular nodes. This could be node strength, node efficiency, node clustering. We can then take one step further and move to connection level comparison. This is the most common approach in connectomes. This is where we're making inference about connectivity strength, whether that be the streamline count or functional connectivity. And this is what I'm going to focus on in the remaining part of this presentation. What we would want to do here, if we're specifically looking at connectome inference, we can go down the path of connectome-wide association study at CWAS. This involves testing our model. So this could be a t-test, f-test, whatever the particular model is, independently at every connection in our connectome. And so every connection is endowed with a t-statistic and an uncorrected p-value. The challenge then is dealing with the massive multiple comparison problem across the family of edges. So how would we do that? There's generic methods to do that. So one school of thought is, well, we don't need to correct for multiple comparisons. I don't think that's a good idea. It risks the problem of false positives. Another approach is Bonferroni. In general, it's too stringent unless we have really large sample sizes. A good approach is the false discovery rate. This can be computed efficiently. It just requires us to order p-values and apply this threshold. And I'm sure many of you have come across the FDR already. However, These generic methods don't take into account connectome-specific attributes. We know that in connectomes, effects of interest are more likely to be confined within subnetworks, interconnected subnetworks, as I've shown here in the example of scenario 2, where we see that the red connections are the effect of interest and they are clustered in a network. And so there's a lot of evidence of this in pathology. I've shown some references there on the right-hand side. We also see this in terms of behavior and cognition, that the effects of interest are clustered within networks. So scenario 2 is more likely relative to scenario 1, where the effects of interest are randomly or sporadically distributed across the network. Now, from the perspective of FDR and Bonferroni, both scenario 1 and scenario 2 are equivalent. There's no difference for them. But there have been methods that have been developed that leverage this connectome-specific attributes, this subnetwork-like behavior, to improve statistical power. One of those approaches is the network-based statistic, the NBS, and this involves the same process of testing our model, our statistical hypothesis at every edge, and then we threshold those edges. So we only keep the edges that are above a minimum effect size. And then in those thresholded set of edges, we aim to identify subnetworks. And so in this example here, I've shown a subnetwork that was found, and it comprises 5 connections. And then we can do permutation testing to ascribe a p-value to that subnetwork, which tells us what's the likelihood of finding a subnetwork of size 5 just as a matter of chance. In other words, under the null hypothesis. So just to recap, then the idea is we once again we test our hypothesis at every edge. It is done independently in the sense of mass univariate testing. We apply a threshold to identify the edges with a minimum effect size, and then among those edges we seek to identify connected components. And in this example, we find a component of size 5. And then permutation testing, as I've shown here, can be used to ascribe a p-value to that component. This involves shuffling patients and controls, and then repeating this procedure again and again and again to build up an empirical null distribution. So here's an example of an empirical null on the right, and we can use that to to estimate a p-value. There are a range of other methods for performing CWAS. I've listed some of these here. This is certainly not an exhaustive list, and I'm not going to have time to go through each of these. These generally vary in at what scale the null hypothesis can be rejected. So are we performing inference at the level of connections, subnetworks, or the whole network? And some of these methods also start to move into the area of machine learning and prediction, particularly the regression. So there's a bit of a grey area here between inference and prediction. I want to now move on to prediction and using the connectome to predict, for example, outcomes, individual outcomes, treatment response, and diagnostic status. And this involves cross-validation using test and training sets. So what we can do if we've mapped our connectome for each individual, we can take the upper triangle, remember these matrices are symmetric, or even the lower triangle. We can vectorize that and come up with a feature matrix where we've got subjects by features, and then divide that matrix up into a test and a training set. And once we have that format, we can use generic supervised learning methods to train a model to predict our desired outcome. And so Dady and colleagues have used this and evaluated predicting binary outcomes in various data sets. So the horizontal axis here represents the improvement in the AUC. So the more to the right we go, the more improvement we have in AUC. And what we see here is that most linear methods perform quite well. So whether we're using support vector classification, ridge regression, or logistic regression, we get more or less the same performance as long as we impose L2 regularization when we're training the model. Related approach is referred to as connectome-based predictive modeling. This is where we use a very simple model. We just got it's effectively a correlation that we use, and we pick out features to that yield a strong correlation in terms of cross-validation. I won't go into the details of that here. Just to wrap up, to give some tips when we're performing connectome-based prediction, If you're using functional connectivity matrices, as has been shown by Varoqai, it's useful to map the correlation or the covariance to a tangent space. This provides, I guess you could think of it as a more natural home for covariance, and this can improve prediction accuracies, as I've shown here, although the improvement is relatively small. It's also important to remember that error bars on accuracy estimates can be large, particularly for small sample size. And this is an argument. If we're going down the path of connectome prediction, large sample sizes are important. Here are some more general tips for prediction. So in particular, k-fold should be used over leave one out. Area under a curve is a good measure for accuracy evaluation. Be cautious of data leakage between test and training sets. And if possible, it's good to evaluate your train classifier across different sites. And this allows us to investigate how well the classifier, how well the model generalizes across scanner sites. So to wrap up, here's some software that you might want to consider using if you're performing connectome statistics. The BCT is perhaps the most relevant. There's both a MATLAB and a Python version of the BCT, and I've provided the websites for both those versions. And once again, this is not an exhaustive list of software for performing connectome statistics. So finally, I just wanted to draw your attention to the Australian School on Connectomics. This is a school that introduces the fundamentals of connectomics, not only statistics, but also mapping and connectome analysis. We're running the next school in Melbourne in 2021, so keep your eyes open for the advertising. If interested, consider joining. Further reading on the topics here, take a look at chapters 10 and 11 in our book. This is with Alex Fornito and Ed Bullmore. And finally, as I've already mentioned, the slides, all the slides that are presented here are available for download from the website that I've shown here at the bottom of the slide. So thank you very much, and I hope you enjoy the rest of the workshop. Thank you.
Hi, thank you very much for joining this part of the education session. Many thanks again to Alex and Andrew for organizing this wonderful educational workshop. I'll talk today about communication and brain networks. So you've heard today already about how we can describe the architecture of the brain. What we believe that architecture is ultimately used for is to transmit signals in the form of electrical impulses between neuronal populations. So we think that there is a myriad of signaling events that takes place on this anatomical foundation, and the superposition of these elementary signaling events ultimately manifests as patterns of interregional covariance that we call functional connectivity. So obviously, the two extremes on the right, structural connectivity and functional connectivity, are two domains that are empirically accessible to us, but the nature of the communication process that links them is something that we have to infer from the data that's available. And what I'll talk to you today over the next over the next 30 minutes or so is how over the past 10, 15 years, different groups have tried to tackle this problem. And really, it comes down to a number of approaches. One is to simply ask what type of communication process is the most biologically plausible, given what we know about neuron biology, physiology, and so on, and brain anatomy. Another is to ask what type of communication process could actually take advantage of the unique architecture that the brain has and all the properties that you've learned about in the other talks today. And finally, because we think that patterns of functional connectivity arise from communication processes, we can actually ask what types of communication processes actually make the best predictions about functional connectivity given structural connectivity. And we can use this to disambiguate and select the most plausible models. And so without further ado, the way that I'll discuss the topic today is to really try to summarize the many ways that people have thought about communication taking place in the brain. One simple way to organize these different models is along an axis that goes all the way from highly decentralized diffusion-like mechanisms all the way to highly centralized routing-like mechanisms. And that's the dichotomy that I'll focus on today. On the one hand, with routing-like mechanisms, we have we can have configurations where a single signal is emitted from a source node and it finds the optimal way to the target node, so-called shortest path routing. That is really the foundation for many graph theoretic concepts and measures. On the other extremes, we have many communication mechanisms that are related to the concept of a random walk. So a signal is emitted from a source node and it is then randomly forwarded to nodes that are anatomically connected. And we measure the probability of that signal eventually reaching the target node. And interposed between these extremes, there exist many different types of communication mechanisms, some that require a little bit more knowledge about network architecture, some that require a little bit less. And indeed, this axis is one of information. How much information would we need in order to get the communication process to actually take advantage of the underlying structure? So I'll start at this extreme, focusing on shortest path routing and communication. If we think about the underlying system that we're studying, we're really talking about action potentials that are transmitted from neuron to neuron in the form of electrical impulses and ultimately through electrochemical synapses. This process is inherently slow. It's a physiological process that takes time. It is noisy because we rely on messages getting through electrochemical synapses. And it is one that requires a lot of energy to constantly depolarize and repolarize membranes, and also requires a lot of material. So for instance, to package a neurotransmitter into vesicles and so forth. So if we were to think about what would be the most optimal way to transmit information in the brain, we would try to pick a mechanism that minimizes the number of synapses. Therefore, a really good, very sensible way to start thinking about communication in the brain is from the perspective of shortest paths. So I just want to start out by disambiguating two concepts, and that is the idea of a walk and the idea of a path. With a walk, we take a source node and we take a target node. And a walk is really any contiguous set of edges between these two nodes. So for instance, it can be the following sequence that's indicated in red that actually has a length of 5. And there are a number of such walks that you can envision for this combination of source and target node. A path, by contrast, is a sequence from a source to a target that is minimal. So it's the minimum sequence of edges that you would need to get from the source to target. So it would be something like this, where you go through these 3 edges from source to target. If we consider this kind of framework, we can measure a lot of important properties of individual nodes and edges, and also of the network as a whole. So we can ask, for instance, what is the mean path length from node I to other nodes in the network, the so-called closeness? So how easy is it for a node to influence its neighbors and for other neighbors to communicate with that node? We can ask what proportion of shortest path in the network actually go through a particular node, the so-called betweenness centrality. If a node sits at the intersection of many different shortest paths, then that node is presumably well positioned to act as a bridge or as a way station for signal traffic. Finally, we can look at how the network as a whole is optimized to transmit information. So probably the most fundamental concept that we have here is the idea of the characteristic path length. So what is the mean path length between all possible pairs of nodes? If this number is very low, or correspondingly, if the global efficiency is very high, then this network is well organized to transmit information from any node to any other node on average. And having low path lengths and high or correspondingly high efficiency means that the network has greater communication capacity, that there is lower potential for signal attenuation. And there have been, over the past 10 or 15 years, many studies that have shown that shorter path length is actually associated with greater cognitive capacity, with greater cognitive engagement, that it's concomitant with normal neurodevelopment and so on. So having low path lengths appears to be a desirable property of the brain. And this really constitutes the first piece of evidence that maybe shortest paths are very important in brain network organization. What I want to show you now are two additional arguments for why shortest paths might be important for brain networks. One comes from the idea of small world networks. So you heard earlier in the workshop that small world networks, and an example of which is the brain and various types of nervous systems, have path lengths that are nearly minimal. They're very close to the theoretical minimum that you have with randomized networks. The fact that we find that most brains reconstructed with different techniques and at different scales are actually small world networks suggests that maintaining short path lengths is a desirable property. The other piece of evidence comes from the studies on what are called minimally rewired networks. So these are networks where the authors take real brain networks, in this case, for instance, the macaque connectome and the C. elegans connectome. They take the nodes and place them in their original positions, but they start adding edges whereby they try to minimize the total wiring cost of the network. So they try to add edges such that the total length of all the edges is as low as possible. And in doing so, they create these minimally rewired networks. What you can do, and what has been shown, is that when you go through this process, and the original networks here are shown in blue, the y-axis shows the total wiring cost of the network. When you go through this process and create these minimally rewired networks that are shown in red, you can actually drastically decrease the total wiring length in the network. So there is a lot of room for improvement. However, this actually comes at a price, because the average path length in these networks actually increases. So in other words, maintaining low path length is a costly feature, but one that appears to be readily incurred by brain networks. And I think these two kind of arguments really really emphasize this idea that maintaining minimal path lengths is something that's very important in brain networks. So you have I've shown you a few arguments for why shortest path routing might be important in the brain, but there are actually many more arguments why it cannot be the complete picture. So when we assume shortest path routing, we're actually making two critical assumptions that might not be biologically plausible. One is that network elements, so for instance, neurons or neuronal populations, actually know the global topology. They know what the architecture of the network is, and they can actually compute the shortest path from any network element to any other network element, which is difficult to envision. The other assumption that we make is that really only a small proportion of the connections will ever be utilized. So just to illustrate this example, here is one network that's been reconstructed from diffusion weighted imaging. It's got 1,000 nodes by 1,000 nodes. This adjacency matrix now is shown on the right, where I'm only showing you connections that participate in at least one shortest path in this whole network. What you can see is in the original network, we go from about 24,000 connections to only about 6,000 connections in this shortest path skeleton. What this means is that if we assume shortest path routing, we're saying that only about a quarter of the total infrastructure is actually being used at any given time, and the rest is really just vestigial. In the field, we've started to think about, could communication occur along non-shortest paths, or alternatively, could we think of communication mechanisms that do not involve the concept of routing? And so one simple way to extend the idea of shortest path routing is to focus on what are called path ensembles. So not to think about just the shortest paths in the network, but to think of the top k shortest paths that are nearly the shortest paths to go from a source to a target. And so this is the idea of path ensemble and k shortest paths. When we talk about k shortest paths, we are really asking what are the top k near minimal paths to go between a source and a target. So we're going from the idea of there being just a single communication channel between two nodes, and we're really opening up, if you can think of it this way, we're opening up an entire spindle of communication channels where there are multiple ways to go from a source to a target. And the panels here, A through D, just show different examples of these top k-shortest paths for different combinations of source and target nodes. The big advantage of this kind of way of thinking is now it allows us to consider accessing alternative or near optimal paths. And in doing so, we are now thinking of a communication mechanism that's much more resilient than shortest path routing, because if damage were to occur to any one communication channel, there are alternative ways to get from any given source to any given target. The other reason this is important is that it will allow, obviously, more network connections to contribute to the overall communication process. And just to illustrate this idea, this will really drastically change our view of what are the most important nodes and edges in the network. So this is just an example showing what are the most central edges and what are the most central nodes in a brain network when we consider either K1, so just the shortest path, K50, which is the top 50 shortest paths, or K100, which is the top 100 shortest paths or walks, if you will. The connections and areas that are shown in green or teal are those that are important just for K1, and the ones that are shown in orange are the ones that are most central for K50 and K100. What you can see is that the actual connections and the actual areas that start to pop out are very different when you start to broaden your horizons and start to consider these near optimal shortest paths. So even something as simple as what are the hubs in my network will drastically change depending on how I think that communication should take place. And this is an idea that I'll come back to in a few minutes. One way that we can actually generalize the idea of path ensembles is not to consider just the top 50 or the top 100 paths, but to really consider all possible walks between a source and a target. And that brings us to the statistic of communicability. So how can we actually quantify the total communication capacity between two nodes without actually trying to say, okay, I want to consider just 50 or 100 or 1,000 paths between them? Communicability is a statistic that actually is a weighted sum of all possible walks between a source and a target. And it's one that weighs the short walks more and the long walks less. So it emphasizes more direct communication channels than the less direct ones. So just to give you an idea of how this statistic works, we pick a source and a target. And the communicability between between nodes I and J is defined as the sum of the ratio of the number of walks of length n divided by n factorial. So for instance, the number of walks of length 1 between these two nodes is 0. So we have 0 divided by 1 factorial. The number of walks of length 2 between these two nodes is 1. There's only one walk. In this case, this is the shortest path between them. So we have 1 divided by 2 factorial. If we consider walks of length 3 now, there are actually 2 such walks shown here in green and also here in green. So we have 2 divided by 3 factorial and so on. The reason why communicability is defined in this way is because this essentially forms the Taylor series. So this entire sum can be written as the matrix exponential, where A is the adjacency matrix of the network. So it's a very elegant way of weighting the short walks and the long walks in a way that can be very efficiently computed in essentially just one line of code. Communicability now brings us to the idea that we want to consider not just shortest paths, but really all possible ways of communicating in the network. And so I'm going to now shift our attention to a related concept, which is the idea of random walks. And now we're all the way on the extreme of diffusion. So when we consider random walks, really what we're talking about, and I think most people are intuitively aware of the concept, is that we have an agent that we drop in our network who probabilistically moves through the graph. So the agent, for instance, here is in the black node, and the agent at any given time, at any given step, looks at the neighboring nodes. So for instance, the agent here has 4 neighbors and chooses randomly where to hop next. So for instance, in the case of a binary network here, the probability of moving to any one of these nodes is 1/4. So the agent, for instance, will randomly choose to move to the neighbor on the right. And the way that this process actually is implemented is we take our adjacency matrix and we compute what's called a transition probability matrix. So we normalize the rows and the columns so that they sum to 1. And so if we have an agent that's sitting at node I, we look at its position at node I in the transition probability matrix, and the elements of this entire row give us probabilities of moving to any one of the neighbors. So if the walker chooses to move to that particular node, we simply move down in the transition probability matrix, and we recompute the probabilities of moving to any one of the neighbors of that node. And the process can continue in this fashion. The main thing that's important for this process is that the random walker will ultimately communication pathways for our network. By considering random walks instead of shortest paths, we can actually compute a number of measures that are analogous to what we had before with shortest path routing. So for instance, we can compute what is the expected time for the walker to travel from node I to node J, the so-called mean first passage time, which is a measure of how easily two nodes could communicate. We can ask what is the minimum number of walkers that I would need to generate at node I in order to guarantee to within some degree of probability that at least one of them will reach node J, the so-called resource efficiency. In other words, how strongly do I need to broadcast my signal in order to guarantee transmission at a particular target node? We can also ask what is the expected time for a walker starting at node I and a walker starting at node J to actually meet on the network, the so-called coalescence time. So how easy is it for signals to become integrated if they start at 2 different parts in the network? And the nice thing about this kind of framework is that it is infinitely extensible. So now we can introduce biases to nodes where they move towards certain types of, to the walkers where they move towards certain types of nodes. For instance, nodes that have certain types of psychoarchitectural properties, morphometric properties, and so on that belong to certain parts of the certain intrinsic networks. We can impart memory on the nodes, on the walkers, so that we ensure that the walkers don't double back to where they've been before, where they're a little bit less likely to go. We can introduce different forms of interaction between multiple walkers in the network, mimicking functional interactions and so forth. And so it's a very powerful framework. The key concept that I want to get across today is that it is actually possible to think about the idea of random walkers finding and looking for shortest paths in their network, taking advantage of the shortest path architecture. And so what I'll spend the next 5 minutes or so talking about is the idea of search information with path transitivity. So with with search information, we are talking about a statistic whereby we want to quantify how easy is it for a random walker to actually find a shortest path between source S and a target T. So what you're seeing here is two different shortest paths, one that is kind of a straight shot from source to target, and one that goes through a series of highly connected nodes or hubs. The idea here is that a signal that has to go through a series of highly connected nodes is more likely, if it's a random walker, is more likely to leave the shortest path to get lost. Or in biological terms, a signal that starts in a particular node and goes through a series of highly connected areas is much more likely to become interrupted or to be subject to noise and interference from other signals in the network. So the statistic of search information is a measure of how much information would you have to impart to a source, to a signal, in order to ensure that it gets from a source to a target. And it really comes down to a function of the degrees of the nodes that you encounter along the way. Because the greater the number of high degree nodes, the greater the degrees that you encounter along the way, the more information you have to keep giving to your random walker in order to ensure transmission. Related statistic is path transitivity. With path transitivity, we are counting how many transitive closures there exist along our shortest path. In other words, how many triangles there are. Because if there are a lot of triangles, that means that even if our random walker were to leave the shortest path, they could still get back on the shortest path because of these triangles. So it's in biological terms, it's a measure of how much does the architecture help you to preserve the signal and potentially amplify the signal as you go from source to target. So what the authors did in this study, this is by Gagne and colleagues, is to take a structural network and compute the search information and the path transitivity between all possible pairs of areas. And that's what's shown here in the upper triangles. What they then said was, okay, we want to know how well does this recapitulate patterns of resting state, fMRI functional connectivity. And that's what's shown in the lower triangles. What they showed was that, indeed, the greater the search information between two areas, in other words, the greater the degrees that you encounter along the way, the greater the chances for the signal to be corrupted along the way, the lower the functional connectivity between a pair of areas. Conversely, the greater the path transitivity between two areas, the greater the number of transitive closures, the greater the potential for the signal to be preserved and reinforced along the way, the greater the functional connectivity between them. And what I think this study beautifully illustrates is 2 points. First, this idea that we want to use the relationship between structure and function to look at how well different communication mechanisms actually perform. And two, this idea that shortest path routing is different from the idea of shortest paths as an architectural feature. And we can, in fact, think about other non-routing mechanisms that potentially take advantage of the underlying shortest path architecture. So we can now actually think about how well do routing and diffusion potentially take advantage of brain networks, and how well could they potentially play out on different types of architectures. So one way to address this question is to use what are called synthetic networks, where you synthetically grow networks to optimize different properties. In this case, the authors grew different types of networks to optimize either routing efficiency, as shown on the y-axis, or diffusion efficiency, that's shown on the x-axis. And when you do that, you create what's called a morphospace. So this is a space of different types of network arrangements that are possible given the constraints. And the nice thing about this, this kind of paradigm is that this morphospace actually starts to form one or more fronts. So these are frontiers of what kinds of network arrangements are possible given these constraints. And what you can clearly see is that you have, first of all, multiple fronts, but in front one, you have networks that clearly have small clusters of nodes, that have modules, that have prominent community structure. And in these networks, routing efficiency is actually better than diffusion efficiency. And the reason for this is that if you have community structure, if you have the existence of clusters of nodes that are densely connected with each other, but have relatively few connections with the rest of the brain, there are relatively few opportunities for random walkers to escape these modules. Communities or modules trap random walkers, and this is something that you'll definitely hear about in the talk on modularity and community detection. So routing is a much better mechanism for networks that have prominent community structure. In front 3 here, what you have is a very different type of network, and these are networks that have a very prominent core or rich club. So this is a central group of nodes that are densely interconnected, whereby, and here, diffusion efficiency is not actually disadvantaged relative to routing. Why? Because a signal that starts anywhere in the periphery could, just by chance, move into this connective core, and in doing so now has access to the rest of the network. So it can easily get from one part of the network to another because of the central component. So this is very interesting to us because, as you've heard in the other talks in this workshop, is that brain networks actually have both a prominent community structure and a prominent connective core or rich club. So the big question is, which communication mechanism actually works best for brain networks given this type of result? And I'm going to give you a minute at home to think about this, but fortunately, the authors did address this question. So what you're seeing here are different types of naturally occurring networks, including brain networks. They're plotted in this graph showing differences between routing and diffusion efficiency. And what you can see clearly is that most brain networks actually lie very close to the identity line. In other words, because brain networks have both a prominent community structure and a connective core, they can actually balance and potentially support both routing and diffusion efficiency. And this illustrates another important point that I want you to take away from this talk, and that is that the nature of a communication mechanism cannot be considered in isolation. A communication mechanism must be thought about with respect to the architecture that it actually plays out on. And different types of communication mechanisms can be advantageous for different types of networks, depending on what properties those networks actually have. So finally, I want to just talk briefly about the idea of percolation models, which take the idea of random walks in diffusion, and but with a little twist. With random walk models, what we are assuming is that the number of signals or walkers is conserved. So if I drop one random walker, that walker will walk around forever. If it's 2 or 3 or more, there will always be that many walkers. With percolation, we're really talking about a process where a signal actually starts from a particular source node and then emanates and is broadcast along multiple fronts and can actually reach multiple parts of the network. So depending on the field that you come from, this can either be called a voter model or percolation or a linear threshold model. I'm going to refer to it as percolation. The idea is that we can have a signal that originates in a particular source node, and the only rule for the dynamics here is that some proportion of a node's neighbors have to adopt the signal before the node actually adopts the signal itself. So for instance, the 2 neighbors of this node have 3 neighbors each. So from their perspective, a third of their neighbors have the signal which is right at the threshold. So both of them would adopt the signal. Now looking at their neighbors, for some of them now the threshold is exceeded, the 2 nodes here at the top. The node here has actually 4 neighbors, so only a quarter of its neighbors have adopted the signals, which is less than the threshold. So this node will not adopt the signal, and this perturbation then is diffusing along multiple fronts and forms a signaling cascade through the network. So we can imagine how that would play out on a brain network. And the nice thing about this type of model is that, again, we can choose different types of extensions where we think about a signal or a perturbation that can, for instance, represent the transduction of a sensory stimulus. We can think about multiple sensory stimuli generating signaling cascades that synergistically move through the network, so more of a competitive cooperative scenario. Or we can endow these signals with different content, and we can ask how they spread through the network, and ultimately where do they form fronts and where do they integrate in the network. So we can ask questions such as which projections are being used to transmit these signaling cascades. And we can also ask what are the most highly integrative areas in the network. So in this particular study, one of the main findings was that the areas that we typically think of as being polysensory or transmodal in nature also tend to be the areas that are likely to sit at the intersection of multiple signaling cascades, showing that the structure of the network actually gives rise to a lot of this integrative character of these in these polysensory areas and networks. Now, I just want to talk for the last 2 or 3 minutes about some new frontiers in this field. One exciting frontier is the idea of navigation. And here, I just want to mention that with routing and diffusion, they're both fundamental concepts, but neither is completely satisfying as a biological mechanism. With routing, for obvious reasons, because we do not want to necessarily think about neural populations having knowledge of network topology. With diffusion, we'd like to think that maybe there's some more order in how signals are being transmitted. Navigation is really a decentralized mechanism that I think allows us to take advantage of the infrastructure in a more biologically realistic way. So with navigation, what happens is that you transmit signals from a source node to a target node, but you do so without knowing the network topology. What you do know is your position and the other node's positions in some other underlying metric space. So for instance, this metric space could just be the positions of the nodes in 3D Euclidean space. And what this example from Kaya Sagan and Andrew Zaleski showed was that if you have a navigation mechanism where signals are forwarded to nodes that are physically closest to the desired target, you can actually recapitulate the majority of the shortest paths in the network. So that's what's being shown here. The success ratio is the proportion of shortest paths that can be recovered by blindly forwarding signals to nodes that are closest to the desired target. And the x-axis is just slightly different reconstructions of the network. The curve in red is what you get when you do so on the real brain network. And the curves in green and in blue are 2 different types of null models. What the authors showed was that indeed, you can recapitulate nearly 100% of the shortest paths using just the simple decentralized form of communication. And indeed, you can use this kind of navigation mechanism using other types of annotations and metric spaces. For instance, select architectural similarity, embedding in large-scale gradients, hierarchies, and so forth. And really, this kind of mechanism is really optimal for spatially embedded networks such as the brain. I just want to show one slide before I finish about how some of these concepts can be really adapted to other problems in neuroscience and engineering more generally, and medicine. One is that we are increasingly using these types of concepts and measures to look at the effects of brain stimulation. So brain stimulation, such as transcranial stimulation, for instance, and more invasive deep brain stimulation, is increasingly used to treat many different types of diseases and to enhance cognitive performance. We know that it works, but we don't know how. What we do know is that if you stimulate a particular area, you're not going to stimulate just that area. You're going to stimulate the areas that it's connected with and the areas that they're connected to. So the architecture of the network is going to condition the effect of that perturbation. So you can take a brain network and simulate the application of a perturbation and use these types of models and measures to to try to predict how the perturbation will spread through the network and what the effects of that perturbation are going to be. So this could be used to identify optimal targets, to look at how best to apply these stimuli, these stimulation protocols while minimizing the deleterious effects. The other way that we can use these kinds of models is for questions of neurodegeneration. So I've talked about communication from the perspective of electrical signals traveling on brain networks, but we also know that the white matter architecture of the brain supports the transport of molecules that are needed for growth and repair. And we increasingly recognize many neurodegenerative syndromes, from Alzheimer's to Parkinson's to ALS and so on, as being molecular nexopathies. So diseases where normal proteins misfold and spread via brain networks. And what's really happening in these types of diseases is that these normal proteins misfold and cause healthy proteins that are shown here in gray to misfold themselves, and they spread transsynaptically from cell to cell like an epidemic. And when enough of these misfolded proteins accumulate in a cell, they cause the cell to die, and they ultimately cause atrophy in the brain. So the architecture of the network can be used to predict the course and the expression of these neurodegenerative diseases. And what you can do is take patterns of structural connectivity and try to predict patterns of atrophy that we measure either from T1 weighted MRI and using various morphometric techniques, such as reductions in cortical thickness, deformation-based morphometry, and so on, or using PET, for instance, if we have tracers for tau amyloid and other misfolded proteins. We can model the spread of these misfolded proteins on white matter networks as a disease or an epidemic that spreads through these networks, and we can try to predict what brain areas are going to be most vulnerable to cell death and atrophy, and actually compare these predictions to real data. So these are kind of two different, I think, frontiers for this kind of work, looking at brain stimulation and manipulation and network transport. The last slide that I want to show is that the assumptions that we make about communication processes are actually very determined to how we assign importance to network elements. What I just want to show is one very stylized network and a quiz for the viewers at home. And that is, what do you think the most important node for communication is in this network? I'll give you a second to think about that, but the answer is that there is no right answer. What you need to know is how communication takes place on this network, because if you think that communication takes place just by forwarding signals to your closest neighbors, you might want to pick the node with the greatest degree, the one with that participates in the greatest number of connections. If you think that communication takes place via shortest paths, then you might want to pick the node that sits on the greatest number of shortest paths, in which case you would pick the node with the greatest betweenness. And finally, if you think that communication takes place using all possible walks and is kind of simultaneously broadcast through the network, you might want to look at the node that has the greatest communicability with the rest of the network. So even something as simple as picking the hub of this network becomes a very complex problem when you have to start thinking about the communication mechanism that takes place. In other words, you might be agnostic about how communication takes place on your network, but the network theoretic statistics and measures that you pick to describe your network, to describe your nodes, are not agnostic, and you really have to be aware of the assumptions that they're making. So just to conclude then, I want to, first of all, say I hope I've convinced you that all network measures are assuming a very specific communication mechanism, and that you must consider the assumptions that they're making, their biological plausibility, how well they can make predictions about real data, whether it's functional connectivity or patterns of neurodegeneration. That shortest paths are a critical substrate. They're a property of any network. But shortest path routing is not the only possible communication mechanism, despite the fact that it's kind of the default measure of many graph theoretic measures. And we can actually really think of a myriad of other very interesting mechanisms that could play out on these networks that we're getting from brain imaging. That communication depends on the properties of the underlying network. It is not enough to just consider the communication mechanism, but really to think about what kind of network it's actually going to be used on. And whether we are talking about modules or cores or spatial embedding, all of these properties are going to influence how well a communication mechanism works. And finally, I did not talk about this in great detail, but really there could be multiple mechanisms that are operating simultaneously and are multiplexed in some fashion, which is something that we typically do not consider. And then finally, that a lot of these very complex questions are actually occurring in the context of multidimensional trade-offs, where we're thinking about the efficiency, the cost that it takes to maintain a communication mechanism, how much information would be required to impart to a given signal, the resilience of these communication mechanisms to damage and so forth. So on that note, I just want to thank you for your attention. Thank again, Alex and Andrew, for organizing this, and to the folks at HBM for really putting on this alternative format for the talks. Thank you very much.